Okay, so I will go ahead and start recording for the last, when are we done? 40? Okay. So our last 15 minutes we can kind of talk about general stuff. One of the things that I think is kind of useful to get at is realizing that biochemistry is this massive, massive subject area. And at least when I went through and took it, I was kind of bored with it just because there's a lot of, to me, biology. And to me, biology was, here's a fact, memorize it. Here's a new fact, memorize it. Ready for the next subject? Here's a new fact, memorize it. And what happens when the test comes? You just spit all that information right back out. Okay? There's a lot of that in this class. Okay? It's different from, say, something like organic, which I'm sure people will argue with me back and forth on. Organic requires a lot of application of knowledge to individual sections. You don't get a lot of application within each of the individual sections of biochem. Okay? What you do get a lot of is, here's organic chemistry. How does organic chemistry apply to biochemistry? Okay, so everything that works in biochem works because of organic chemistry. Okay? Now everybody's saying, oh crap, I hated organic chemistry. There's a difference in that application process. Biochemists tend to have a very poor understanding of organic chemistry. Most of them insist that there's no organic chemistry whatsoever in biochem. Okay? Which is mind-boggling. Okay? But the reason why is an organic chemist went in and said, this is how it works. And the biochemist said, oh, okay, now I can teach it. This is how it works. Well, why does it work? Well, I don't know that. I don't need to know that. That's not biochemistry. Oh, screw off. Okay? So we'll keep coming back to these organic chemistry concepts in kind of a simplified fashion. The most common things you'll see will be functional groups, intermolecular forces, and to a certain extent, some reactions, okay? But to a very limited amount. Okay? What actually got me inspired or kind of interested in biochemistry as a field was this fancy molecule, which some people might be able to guess what it is. I don't think I put the name up there. What was it? Not quite. It has to do with ATP, but it is not ATP itself. ATP is adenosine triphosphate, relatively small molecule. Okay? This thing is massive in comparison to ATP. ATP, as far as scale goes, if I can get this figured out here, if we draw ATP in there, I think it sits right there. See that little blue dot I put in there? That's roughly the size of ATP. Okay. No, glucose is even smaller than ATP. What is that? No. This is the protein that synthesizes ATP. This is ATP synthase. Okay. Fascinating molecule. Really interesting, rich history. It's got all sorts of fun debates about it because this is effectively, for all intents and purposes, an engine. Okay, it is the smallest engine that we know. Okay? Why is that kind of a fascinating, interesting subject? Because we can take all these molecules and we can generate these cool energy. We can generate fuel. It does all sorts of rotations and movement and all this fun stuff. What makes that even more fascinating? If you're a creationist, this is the reason there's God. Is this molecule. So this molecule, or this protein, keeps coming back to this whole creationist theory. Okay? So there's a lot of neat movies and a lot of neat studies that come out of using ATP. So I'm going to go ahead and pull up, in theory, I'll be able to pull up here a movie. See if this actually works appropriately. It probably has a commercial attached to it. No, it doesn't. So we've got an animation of ATP synthase actually doing its job. So that giant ball that we saw at the bottom, that's this guy up top. You'll start to notice that everything looks globular and a big mess. Well, that's because it's a globular protein. That's what it looks like. Okay, now what we're going to do is zoom in at an individual section. You'll notice there's ADP sitting in there. Where's my phosphate? Oops, no, that's the protein. There's ADP. There's a phosphate. What the protein does, it literally opens up, ADP comes in, it wraps around it, and it forces those two molecules near each other. In the process of forcing them near each other, we're now at the perfect orientation where we can form that bond. No more solution, mix some stuff together and hope it works. The protein goes through and forces it to happen. How does it force it to happen? It's powered by a proton gradient. We're shuttling hydrogen ions across a membrane. As those hydrogen ions flow across the membrane, it causes the whole protein to rotate. As the protein rotates on this axle, it changes conformations up here. Those change in conformations allow for the formation of ATP, for reaching out, grabbing ADP and phosphate and forcing them together to make this awesome structure. Why is ATP even further super cool? Well, it's what powers pretty much everything we do with in life. Okay? So this protein is just kind of a fascinating molecule all around. Okay? How do we know it rotates? Because we're looking at something that's wicked tiny. Good question. We have to design experiments that would allow us to go through and see that, which isn't the easiest process to go through and do, but there are fields where they went through and designed this. They've gone through and they've built, this one's fun. They took the molecule, they got rid of this section of it, okay, and then what they did down here is they fused this end to a surface, so it couldn't move. And then what did they do on the little axle part? They put a gold nanorod, super tiny little gold bar onto the end of that. And then they threw in a ton of ATP. Well, what ends up happening? ATP binds in here and causes this thing to start changing conformations, which then causes the rotor to spin, which then causes the gold nanorod to spin. So we can't see the protein, but we can see the gold nanorod. So what ends up happening? This thing goes through and does its rotations, and if we look with a microscope, what do we see the gold rod do? Start to rotate. Oh, crap, I guess it's rotating. So we can come up with kind of some interesting experiments to evaluate these things. Right? It's kind of fun stuff. So that was one of my first kind of existences into biochemistry, was looking at that and actually saying, okay, this is actually kind of cool stuff. We can do some fun things with it. Okay? So, where does it come from? We've added the comment here that this is the optimist view of it. Right? We've already talked about my background is primarily in organic chemistry. So where do you think our optimist view of biochemistry is going to come from? It's going to be organic chemistry. It's all of our functional groups. Okay? It's how each of those individual functional groups interacts with other functional groups. And it's those interactions that drive biochemistry, that allow it to work and function. So what we need to be able to do is to look at these functional groups and be able to talk about them intelligently, okay, which comes to an interesting idea of where did these fields come from. And when we started biochemists, the first biochemist sat down and said, oh, it's organic chemistry, and started drawing functional groups, right? That's how they started biochemistry? Probably not. They probably said, hey, there's something live, it's doing something weird. Why is that live thing doing that? Now let me look at it further in. Oh, I found a protein that's now allowing that live thing to do this. Well, what is that protein made of? How does that protein work? Okay. Which is an odd concept. When we talk about and think about any of the scientific fields, we look at the very basics and we work up to these big concepts. But how were they actually discovered? The complete and opposite reverse direction. We looked at these massive concepts and said, okay, let's work our way back down. And in the process of working our way back down into those individual sections, we had to come up with rules so that when I talk about it, you understand what I'm talking about. Okay? And this becomes frustrating when we talk about science and organic chemistry and structures. Okay? You throw a random general chemistry student into an organic chemistry class, they have no clue what's going on. Okay? They still understand some aspects of chemistry, but why do they have such a hard time with organic chemistry? They have to learn a new rule set. Okay? So what we're going to try and skim across the surface of are what are those rule sets? What makes up proteins? What makes up DNA? Okay? What makes up RNA? What makes up carbohydrates? So we're kind of looking at this class as the subsets. What are the rules? You kind of have to add into, okay, what are the larger aspects of this? Okay? We'll do kind of breakouts every so often from the material to say, oh, this is where we can apply it. This is how it's going to work. Okay? But realize we're going through these rules that are very boring because it's just a rule. Okay? They weren't discovered first. They were discovered last. Okay? So when we go through and look at these, you do need to know your functional groups. Okay? Any ideas which ones are going to be the most important? Stick.
Alcohols.
Okay, alcohols are going to be pretty important. Why is alcohol important? Okay. We got hydrogen bonding with our OH group. Okay. More important functional groups.
Aldehyde.
Okay. We got aldehydes.
Ketones.
Where'd the aldehyde go? There it is. Anybody know what happens to alcohol when it gets processed by the body before it gets excreted? It gets turned into an aldehyde. It gets then turned into carboxylic acid, which is then excreted by the body through its waste systems. Why is methanol a problem? So if we look at ethanol, 2 carbons, right? If we look at methanol, 1 carbon. So our eth was 2. And for it being all single bonds, and then the all for the alcohol, the OH. First step in ethanol is our CH3, our ethanaldehyde. It's the first step in methanol. Formaldehyde. What's formaldehyde used as? Preservative. Okay. Aldehydes in general are not particularly nice compounds to have floating around in your body, which is one of the reasons why we have headaches or hangovers, is it's a buildup of this aldehyde. Nice process about ethanol. When we form that ethanaldehyde, there's still something that our body can grab a hold of. That extra carbon allows our proteins to wrap around it and ultimately convert that into the carboxylic acid. The acid solubility is significantly higher. Okay, so we turn into, actually here, there we go, ethanoic acid, okay, or acetic acid. Highly soluble in water. Why is it soluble in water? It's a polar molecule due to the hydrogen bonding interactions coming off of our functional groups. Okay? So it's those interactions that are super helpful. When we look at formaldehyde, it's such a small molecule that there really isn't anything for our proteins to grab a hold of very easily. So what ends up happening to the lifetime of formaldehyde in your blood versus the ethanaldehyde? A lot longer. So we now have a toxic molecule floating through our bloodstream for a lot longer lifetime. What ends up happening? You get a lot worse headaches. Formaldehyde is one of the primary reasons why it's a horrible idea to drink methanol. Okay? One of the issues is it can cause death, but the primary one that most people seem to know is blindness. Okay? That molecule gets involved with your ability to see. You were doing optometry, right?
Yeah.
So you know more on this?
No.
Dang.
Yeah.
So we're looking at these molecules not being able to process. Okay? And those things are then going to be what destroys our other processes. So we're looking at not just these functional groups, but then what does your body do to them when it hits the bloodstreams of those individual proteins? Okay? So we just did a completely random side topic on ethanol, but hey, why not? Said aldehydes, said ketones. In fact, I'm going to change the coloring on that aldehyde. Said ketone. Heard an amine show up in there. We got amines. Interesting. I'm now changing the color. Why would I change the color?
Serve a different function?
What?
They serve a different function?
Yes, they do serve a different function. There's another reason behind it.
Because of the amide.
What do you mean amide?
I know that there's even an amide. Amide has a carbon and an oxygen double bond.
What were you suggesting?
It's related to proteins.
What makes up proteins? Amino acids. What does amino mean? Amine. What's our other functional group that's going to be really important? Not just the carbonyl, but the carboxylic acid. Okay. Amino acids, our fundamental building block for our proteins, are composed of two functional groups, our amines and our carboxylic acids. Those two functional groups are pretty much what drive everything we understand of proteins, okay? Or not everything, but a large portion of it. So we'll keep coming back to amines and acids. And you'll notice that the functional groups that we're going to primarily work with are the ones that have a lot of hydrogen bonding abilities, have some reactivity associated with them. So if you think back to your organic chem knowledge, and we think of, say, this middle functional group here, do you think we're going to spend a lot of time talking about that one? Why not?
Not very reactive at all.
Why is it not reactive? Okay. It's going to come back to our intermolecular forces. It's our partial charges that are generated out of this. We have that oxygen. That oxygen has lone pairs. But it is shielded by those carbon structures. Those carbon structures reduce the reactivity of that oxygen to the point that this functional group is virtually unreactive. What functional group was it? We have an ether. Okay. So it's looking at those individual pieces and deciding to play around and approximate them as best we can. Okay. We are definitely done for the day. I apologize.